two highly talented postdoctoral fellows that are in our working group. So this is a Canadian in-house modeling working group that we meet every week. And the two postdocs that are a part of this working group has been working on either COVID or COVID-related projects, mathematical models, basically. The first postdoc, David Dick, will be speaking on the modeling transmission and the impact of waning immunity. That is sort of parallel to the talk this morning by Carol Vignos, but in the context of Canada. Next, Chapin Korosec will be speaking on the mRNA COVID-19 vaccine models and simulations that he has been working on in the last 5-6 months, and also the long-term prediction on the immunity. So without further ado, I'll pass the floor to David. David, are you able to share your slides?
Yeah, I'm just trying to get my slide shared.
Mm-hmm.
Hopefully you can now.
Perfect. Looks good, David.
Perfect. Okay, thank you for the introduction and the chance to share this work. Today, I want to focus on between host transmission of virus, because I know we're looking at both within and between hosts today. And I will look at the pandemic, the SARS-CoV-2 pandemic in Canada. And I'm going to use an age-structured model of SARS-CoV-2 infection. And it's the same model with some minor changes to what Carol presented earlier today. So I'll hopefully just quickly go over the model, but I want to go over it in a little bit of detail. So the parts that are most relevant to what I'm going to show you, I explain clearly. We also want to look at a little bit or a quick discussion about herd immunity. I want to show you some thoughts about the distribution of immunity and a little bit of discussion about the longevity of immunity in the population. So this is the model structure that I'm going to be considering. It's the same model that Carol described to you earlier, like I mentioned, and this is just one, it's an age-structured model with 16 age classes, and here is the diagram for just one of those age classes. Let me get my pointer. So there are a few, I just want to mention there are a few differences in the implementation than what Carol described. So the way we implement vaccination is different in this model. We don't explicitly consider variants of concern, but that's captured in the way our kappa, or the K value that we estimate, changes. And the way we do the data fitting itself is a slightly different genetic algorithm, but it's very similar to what Carol was describing, so I'm not going to describe that in detail. And we can use this model to assess levels of immunity and to build a description. And what we're working to do is to build a description of the hazard rate in the population, or the probability of a resurgence and the probability of an individual being infected based on this immune distribution we have in the population. And it's still very much a work in progress. So I want to just show you what we have as we're working on this. So first in our model, we have susceptible, S1, 2, 3, and 4. And then we have vaccinated classes, same as Carol described. And then we have infected individuals. And the important part for today's discussion is we have different levels of immunity. So S1 are the fully susceptible individuals. And those I indicate in the following plots in red. I apologize that the colors are incongruent. But so red represents the fully susceptible individuals in what we'll see next. And then we also have individuals who have some immunity. So this is represented by the light blue in the plots. And so this would be category S2. So this is someone who has recovered from a mild case of SARS-CoV-2 or COVID-19, or someone who's waned from a more severe case, or someone who's waned from vaccine induced immunity, or someone who's vaccinated from a fully susceptible class with 1 dose of vaccine. And then we have our next level of immunity. So this would be people who have recovered from a more severe, so a symptomatic bout of SARS-CoV-2 or infection, someone who's been vaccinated with 1 dose of vaccine who already had faced a natural infection, or people, of course, who have waned to those compartments from some compartment with higher levels of immunity. So there's always the probability, or there's always a portion of the populations in these classes that have waned back into these classes from levels of higher immunity. And we don't explicitly track people who've been vaccinated and waned back into these classes versus people who have had natural infection. Part of the reason is we already have lots of equations and lots of complexity. And if we wanted to consider all the different routes of how you gather immunity and then how you would wane back to immunity, the problem gets exponentially worse. So we've avoided that so far. And then we have in green, the people who are fully immune. So these people are fully protected from infection. And the other thing I should go back and mention is at each levels of these immunity levels where you have increased protection from infection, you also have increased protection from severe disease. So we assume that vaccinated individuals have protection, an increased protection from severe disease than someone who is, say, fully susceptible. And to parameterize our model, we look at comorbidities. So the risk of severe disease is informed by comorbidities in the population. We use this paper by Knight, Misha et al. to understand the risk of severe disease of COVID-19 based on different comorbidities for different age groups in the population. All the parameters other than kappa, which I'll talk about fitting in a little bit, we estimated from the literature, and we set, so the R naught at the beginning of the epidemic or the beginning of the pandemic is 2.9, same as what Carol explained. Oh, and I see my timer's not working, so I'll try and keep track of the time myself. So as far as calculating the public health mitigation, we also use the contact matrices from Jit et al. And then on top of those contact matrices, we have the mitigation. So we implement a perturbation matrix on top of those to reduce the level of contacts based on changes on public health measures that are announced in the provinces. So Anna has done great work collecting all the public health mitigations across Canada for each of the provincial regions. And we look at the different mitigation strategies in 3 different categories that align with the contact matrices from Jit et al. So we look at school restrictions, and here we have a 4-compartment scale where we have no restrictions. We have schools open, but some restrictions. That would be like school cohorting, social distancing and enforced, that kind of thing. And then we have a blended learning level, and that's where some students are learning from home and some students are in class. And then we have schools closed. And then we have a similar group of compartments for work, where we have no restrictions, businesses open, only essential businesses, etc. And then we also have the same restrictions for other. So that would be businesses, restaurants, that kind of thing. And we collect this data for every province and every territory in Canada. The model I'm showing today doesn't do every province and territory because some of the populations are small for our model to work properly with. And all of this data will soon be available live on a GitHub repository. It isn't currently available, but if you're interested, I think you can contact me and we can discuss it. So here's an example of what the contact matrices can look like between a high level of restrictions. So there's a lot of different combinations of these 4 compartments over the 3 different regions, work, school, and home. So there's a lot of potential mitigation strategies. And this is just 4 examples of it, from high level of restrictions, so very little contacts happening, a lot of people staying home or away from school and away from work, to no restrictions at all. So this is just the base contact matrix that you get from Jit et al. So here's an example of the fit for Ontario. In this case, we're fitting 80% of I3, so these are the more mild infections, but you expect to detect most of them, and all of I4. And if we look at this plot, the red pluses are the actual incidence data. Here we get our incidence data from the COVID-19 Canada Open Data Working Group. It's available on GitHub. It's also available online. And the black line we see in this plot is I4, so the severe infections. The dashed line is I3 plus I4, so the more mild infections and the severe infections, not including the asymptomatic infections, are plotted on this graph. And here we fit our kappa value monthly, so that's what the vertical dashed lines represent. Is inside each of those windows, we fit 1 kappa value or 1 K value to represent those personal changes or those personal masking and other mitigation strategies. And our kappa value also captures things that aren't captured otherwise in the model. So in Carol's description earlier today, they were explicitly considering variants of concern. We don't explicitly consider variants of concern. We allow kappa to capture that change in variants of concern. And then you can just see our kappa value actually can exceed 1 because the variants of concern have that increased transmissibility. Okay, quickly running out of time, so I'm going to skip this slide. And here we see the ascertainment rate that we get from our model. This is from Fitting Canada overall. And I'll just note that here I'm fitting 0.6 of I3 as well as I4. And the reason I want to show you this is because we find in our model that ascertainment rate is affected quite strongly by the probability of severe disease. So as vaccinations start, and especially as they ramp up in April, we see the ascertainment rate go up quite a bit. So we capture a lot more of the virus. That's a lot more of the circulating virus is detected. And then there's also a drop off in this ascertainment rate later on. Some of this I think can be explained because the waning of the vaccinated protection, so you're waning of protection from severe disease, you might get more infection, but I don't think it explains all of it. So there's still some part of this drop off that's unexplained. It could be related to behavioral changes. So people are less likely to get a COVID test if they feel symptomatic because they feel the pandemic's at a different spot. So I think there's a lot of behavioral changes here. And I just want to also mention that that I forgot to mention earlier is vaccine passports complicate our application of those mitigation matrices because if there are vaccine passports implemented, even imperfectly, it'll be the susceptible people who are going to be faced with the burden of disease more severely. Okay, so now let's get to some results in my last 1.5 to 2 minutes. Here we have Ontario. This is the distribution of immunity. So red is fully susceptible, blue has some protection, yellow has more protection, green is fully protected. And I'm comparing waning over 1 year per compartment and 2 years per compartment. And we see a very strong difference. And I'll just mention really quickly, we use the actual vaccination data. The numbers might look a little bit lower than you'd expect because there's waning and because also not everyone who gets a vaccine gets full protection or gets protection from the vaccine. I seem to have used a lot of time. So here I'll just show, I'll go back for a second. Here that we see the burden of disease and the number of susceptibles is shifted towards the younger populations. That of course we expect, we don't vaccinate the younger populations. But here we see the burden of severe disease is still with the darker classes, which is the older age classes. And it's still mostly in both cases, the unvaccinated that are driving the infections, not people who have waned or people who are vaccinated. Now I'll just very quickly go over this last figure. So here in Ontario, at the end of fitting, so end of October, we had about 67% of people had some immunity with waning over 1 year. And if we look at Alberta, before they had their resurgence, it was around 66%. And then of course, with the increased vaccination bump and the resurgence, it goes up to 77%. So just the level of immunity doesn't predict your risk of that resurgence or that new infection level. And if we look at all the provinces now, we see that bump in new infections in Saskatchewan, Alberta, but not in Manitoba and Ontario. And there isn't a large difference in the level of immunity. So that either means Ontario is still at risk or there's something more complicated going on. And with that, I've run out of time. So thank you and I'm happy for questions.
Thank you, David. We probably have time for one quick question if there's anyone from the floor that is interested to. Otherwise, you could still reach out to David either through the chat box or at a later time. All right. Thank you so much, David. Really interesting talk there. We'll move on to the next speaker of the day, Chapin Korosec. Chapin, are you there? Are you able to share your slide, Chapin?
Hello. Yep. Can you hear me?
Yep. Perfect.
All right. So if I do desktop.
try
here.
my slide.
You should all see my slides.
Perfect. The floor is yours, Chapin.
Okay, thank you, James. I need to move the video. That will distract me. So today I'm going to present our work that we have just uploaded to the archive and submitted for peer review 2 weeks ago. It's called Long-Term Predictions of Humoral Immunity After 2 Doses of Pfizer, BNT, and mRNA 1273 vaccines based on dosage, age, and sex. And there's some medRxiv DUI there, and I can provide it in the chat later as well if you're interested. So I'm a postdoctoral fellow in the group of Jane M. Heffernan. So our work concerns the lipid nanoparticle formulated mRNA vaccines. As a really very quick primer, essentially the idea is that we're housed for these vaccines, the spike protein, the mRNA encoding for the spike protein is housed inside of a lipid nanoparticle shell. The shell itself is fairly physiologically inert, doesn't cause a lot of damage. And the idea is that it's going to be injected intramuscularly, and after that, a number of things occur. Because it's a 50-minute talk, I'm not going to go through all of the current literature on how our cells become vaccinated, but really quickly, in a nutshell, what happens is that predominantly most of the dosage is absorbed into our muscle cells. A proportion of it is drained into our lymph nodes, and there's a proportion that's currently thought to be wasted and drains into the liver. And so it remains active. From mouse studies, we know it remains active in the muscle for about a week, at least in mice. And so far, we haven't done a study with fluorescent proteins in humans yet to figure out the human equivalent. But it's the mouse papers that we're basing our models off of. And so the experiments in mice determined that it remains active in mouse for about a week. There's partial, by active, I mean there's spike protein being produced in the muscle cells for approximately 1 week. Partial drainage to the lymph nodes, primarily targeting dendritic cells. Partial drainage to the liver. That we know from mice studies and from human studies, it's activating both vaccines, the Moderna and Pfizer, activating both arms of the immune system. And we know from clinical data in humans that there's a fairly pronounced inflammatory response, particularly through monitoring IF and gamma, as measured by both blood and myocyte tissue sampling. So that's both in mice and in humans with the specific vaccines that we're working with. So our model is as follows. We have a compartment for lipid nanoparticles denoted as L. The lipid nanoparticles are inoculated into the recipient. So this is all within host modeling via some initial condition based on the actual dosage inside each of the respective vaccines. There's an interaction term with the lipids with vaccinated cells. So vaccinated cells we're modeling as V, and there's a degradation term of lipids, gamma L, to take into account those amount of lipids that have fusion inefficiency. So for some reason the payload is not delivered, or there's waste drained into the liver. Vaccinated cells are modeled generally, and so we're not actually specifically going to model muscle cells, dendritic cells, and macrophages. We're going to consider a single vaccinated compartment, and that is predominantly because there's currently not a lot of literature. Well, there's actually no literature that I'm aware of that says exactly what proportions are going to myocytes and activating the immune system, and what proportion is going to dendritic cells and macrophages and activating each of the arms of the immune systems through those different pathways. And so we consider a general approach. T is modeled by T helper cells. And so of course, T helper cells are interacting with MHC2 complexes that are being exposed on vaccinated cells. So naive CD4 plus T cells are given by T in our model, recognize and bond with MHC2, where an information exchange occurs. That's been given by the MUTV term. So they effectively become primed at some rate MUTV, and they have a natural cell death rate given by gamma T. We then model a primary antibody response solely generated by plasma B cells. And so there are two source terms in our plasma B cells in our model. They can, of course, be primed by antigen-specific T helper cells through the interaction term UTB. The maturation of naive B cell into an antibody secreting cell there's a number of literature to support that it's further enhanced by various cytokines. We focus on interleukins because we found some literature to support that. And we model that through the source term given by alpha BI, where in that, I guess you can see my cursor. Oh, it's not showing up for me. So the S subscript I term provides a duplication threshold due to interleukins, so we don't let them run away due to interleukin yeah, we don't let them run away. And then we have a gamma B term just to take into account the natural death rate of plasma B cells. So then plasma B cells being the primary producers of antibodies, so we focus on the production. Predominantly, we're going to focus on IgG because all the data, I found very good data sets for IgG, so we're focusing just on IgG in this paper, and it's given by A in our model. And we don't distinguish between subclasses of IgG in this paper either. And so we model the production of IgG through the source term from plasma B cells. And of course, we have a natural degradation term for that IgG protein as well. And then so we also considered CD8 plus cells primed by antigen presenting cells through cell surface MHC class 1 molecules. And again, we sourced from the literature a number of papers that support IFN gamma, the presence of IFN gamma actually enhancing the priming of CD8 plus cells, and that's captured in that alpha term. Then we have a gamma C term as well, taking into account natural degradation. And so lastly, we have our cytokines, we have IFN gamma, and we have interleukin, and we use various interleukins in this paper. We focus on those interleukins that we find good data for fitting, and so we have a few interleukins that we've used. Predominantly, they're related to the inflammatory response. All of our cytokines are sourced from T helper cells. Okay, so after we developed this model, the idea is that now we're going to fit this within host model to learn about the dynamics of the vaccine and the inoculation, and to figure out some timescales of the vaccination process, as well as we went into this thinking we wanted to figure out the long-term, if we can get a strong prediction on long-term humoral immunity. And so I found a number of papers listed here that focus on clinical data collected in humans, and these are mostly all standard doses. I believe one of these is low dose. Yeah, reference 24 is a low dose Moderna. And so predominantly we focus on standard doses, two-dose standard dose of both Moderna and Pfizer. And we do all of our fitting in Monolix, which does mixed nonlinear mixed modeling effects, and we can actually fit all of these data sets simultaneously. And so what we did is we fit all the standard standard dose data sets together, and we can of course distinguish in our fits between Pfizer and Moderna. And then we fit the two-dose low dose data separately. So there's no nonlinear mixed modeling effects that's in any way affecting the low dose data set. And you can see the population sizes on the right hand column there. So we have, as far as good temporal clinical data is concerned, we have some pretty strong data sets that are going into these fits. And again, we're predominantly using IgG, and we have IFN gamma, and you can see, for example, from reference 19, they focus on a few interleukins, 6, 8, and 15 that we collected and fit to as well. So here's what our fits look like. We have Moderna shown in green, Pfizer shown in red, and we have a population fit shown in blue. So Moderna, all the green fits are fit to all the Moderna data, but they're also affected by the Pfizer fits in the nonlinear mixed modeling framework in Monolix. And what I've shown here is just the outcome of sort of the average Moderna response, and I'm showing the outcome of the average two-dose Pfizer response. And then the population fit is the outcome of all standard two-dose data. you'll notice that all the red points are below the green points. This does not mean that Pfizer has a lower IgG threshold than Moderna, because all of this data, most of this data was done by different hands, and it's all fluorescence-based experiments. And so, of course, if they don't have all of the instrument settings set the same, they're going to have a different y-axis response, and that's why all the data is represented in arbitrary units. However, you can see there's a data set. I'm just going to exit this because my cursor is not showing up for me. There's this data set here that has green squares and red triangles. This was collected from the same source. So when the two vaccines are collected by the same set of hands, the magnitude of the response is actually quite similar. So we'll point that out. However, because all the respective data sets can't be compared on their y-axis, what we can do is actually normalize by the peak response and then compare the decay kinetics between the vaccines, and that we deem as fair game. And so shown here on the top panel is what 2 doses of Pfizer look like for the decay kinetics normalized by peak response, and the bottom panel is 2 doses of Moderna. And the coloring denotes the percent loss milestones. So green, for example, if we focus on the top panel, the green means 25% loss, and that happens by day 79 for Pfizer, all the way down to red, which 99.9% loss by day 265 for Pfizer. And we stop at day 265, which is approximately 8 months past dose 2, because that was the initial timeline for dose 3 set out by the CDC. They have since corrected themselves and are suggesting a 6 to 7 month timeline for dose 3. And as we go through this, I'll go through our results, I think we're in strong agreement with that change. And so just really quickly, what you can see here, for example, if we just focus on the 75% loss milestone, basically we find that Pfizer always decays faster or reaches the milestones before Moderna. And so, for example, 75% loss occurs at day 120 for Pfizer, and that occurs by day 135 for Moderna. And I believe I have a table summarizing everything later. And I'm also going to talk about in a minute the correlation between humoral loss and efficacy, because that, of course, you might be able to lose more than 99% of your IgGs, and you still might actually have a bit of defense. And I'm going to cover that at the end of this, going through the results. So we also, one of our data sets separated their data by age, and because all the age-related data was all collected from the same lab by the same person, we actually, instead of normalizing by peak for these fits, we actually can compare explicitly our predictions as a function of age. And so what we have is age groups 18 to 55, 56 to 70, and 70 plus. And we find that there are very little differences all the way up until around day 100. And then from day 100 to day 250, there emerges a advantage for the younger cohort. And so in the bottom panel of this slide, we plot the ratio of the young cohort to the oldest cohort. And we can see, for example, that there's an initial faster uptake in IgG from the younger cohort. There's a spike such that by day 10, there's a 3-fold advantage. However, that advantage diminishes by day 50, where there's a ratio of around 1 by day 50, after which an exponential advantage emerges for a younger cohort, such that by day 265, there's approximately a 5-fold IgG advantage in a younger person than compared to the older cohort. And just for your information, for each of these age cohorts, there's on order of 10 people that they track through time, to give you an idea of the statistics for this prediction. Yeah, so then next we have data for 2 low doses of Moderna. I wasn't able to find good temporal data for 2 low doses of Pfizer, so we compare the 2 standard doses of Moderna to 2 low doses of Pfizer, each administered at the manufacturer's recommended time scale. I think I forgot to mention that. So all the 2-dose data is at the manufacturer's recommended time between doses. for both Pfizer and Moderna, and both standard dose and the low dose data here. And so it's interesting, again, we normalize by peak response, and essentially what we find is that the decay from peak for 2 low doses is much slower. So for example, 75% loss, oh, there's a typo here. It says they're all day 265. I apologize for that. It's not like that in the table I have coming after this. But essentially what we find for 2 low doses is all the milestones occur much later in time for low doses, so that peak relative decay than compared to 2 standard doses. And then, so here's what the low dose fits look like. And again, oh, so here has the correct, the milestones, 75% loss by day 168 for 2 low doses, compared to 135 for 2 standard doses of Moderna. And then here's the summary of our percent loss milestones, but also our gamma degradation rate that we pulled out for all of our fits. And so you can see Pfizer and Moderna actually have a similar humoral degradation estimate of around 0.04 for Pfizer and 0.038 for Moderna, so slightly faster for Pfizer, but that makes a difference over the course of 265 days. And then we actually find the gamma A to be quite a bit faster for Moderna. However, what I neglected to show here is that the plasma B cell response, the decay or the death rate of plasma B cells is a lot slower for the 2 low doses, or predicted to be a lot slower from our fits. And you can see if you just focus on the 99% column, that 2 low doses of Pfizer is expected to reach 99% by day 209, but then you get an extra 20 days or so for Moderna. It's not until day 231. So in our manuscript, we then go on to discuss some implications of our results, and I'll just quickly go over these and then end my talk. So one of the things that we conclude from our predictions is that regardless of which vaccine one received, assuming the doses are separated by the industry suggested interval, which of course is not applicable to the Canadian scenario. We're working to try to get Canadian data to find fits from a much longer interval, but assuming the doses are separated by the industry suggested interval, we predict that if dose 3 to occur at month 8, the original CDC timeline following dose 2, there exists a period of about 1 month where individuals have less than 99% humoral immunity relative to peak. And then the question becomes, in that month, is it, how much of a concern is that? Yeah, and again, the CDC later revised their plan to 6 months past dose 2. So we then, oh yeah, and this is just reiterating the age-related results, such that for a younger cohort, there's about a 5-fold humoral advantage predicted than compared to an older cohort in humoral immunity by day 265, which suggests prioritizing older individuals is of course important. So then we went on to try to correlate the efficacy loss with our predicted IgG loss, and our model allows us to make a clinically guided qualitative prediction for humoral-based immunologic correlate of prediction. So basically, we're going to correlate the percent loss milestones with efficacy loss milestones to figure out what percent loss relative to peak correlates to what efficacy loss from data collected from countries or data sets where the doses were administered at the company mandated intervals. So one example is that 7 months following dose 2, the Pfizer-BioNTech efficacy drops from 75% immediately after dose 2 to 16%. So this is a recent study done from the Ministry of Health of Israel, and they have very strong statistics for this result. It's a fantastic report that they announced. And so, for example, 7 months past dose 2 for Pfizer, we predict IgG to drop to 0.4% of the peak. So that's a loss of 95.6%. So roughly speaking, 0.5% of peak correlates to something like 16% efficacy, which is quite remarkable.
Chapin, could you wrap it up? We are sort of a little bit over time.
Oh, yeah, sure. And then, yeah, we just do a similar study, and then we correlate, for example, 28 days past. We find there's an increase in efficacy 28 days past dose 2, and we also correlate that Moderna is reaching its peak 28 days past those two. So that matches well with what's found in the literature. And so I'll just end there. Definitely a lot of people to thank that have contributed to this project, from postdocs to NRC research scientists, and definitely acknowledge Jane M. Heffernan as the PI on this project, and Morgan Craig, also faculty at the University of Montreal, who worked on us with this. Thank you.
Chapin, amazing book there. So I'll encourage you to read the preprints that Chapin has put either in the chat box. So without further ado, I'll